Historical perspective. Discovery era, transition era, golden era, era of chemotherapy and microbial genetics, modern era. Discovery era. Robert Hooke. In 1665, Robert Hooke discovered cell using a microscope. He observed cells in a cork slice and noticed honeycomb-like compartments. He coined the term cells for these compartments. Antony van Leeuwenhoek. It is the father of microbiology. First person who observed microbes. They given a term of these microbes called animalcules, fungi, algae, protozoa. Antony van Leeuwenhoek used handcrafted microscope. Firstly observe and describe single-celled organisms. He named them as animalcules, known as microorganisms nowadays. He observed and recorded other microscopic organisms and structures, such as muscles, fibers, blood flowing in capillaries, and bacteria. He discovered bacteria and protozoa. Transition era. At that period, scientists used a theory known as spontaneous generation to explain the origins of life. According to spontaneous generation theory, lifeless objects give rise to living organisms. Examples: simple life form arose spontaneously non-living material. Francesco Redi, it was Italian scientist. It is first theory, who argued over this theory. Challenged the theory of spontaneous generation. In 1748, John Needham, British priest, proposed tiny organisms spontaneously developed on mutton gravy. Edward Jenner, 1798, discovered vaccination for smallpox. He was unable to explain the etiology of smallpox. Ignaz Semmelweis, 1847, observed transfer of infection from autopsies to maternity patients. Showed hand washing could stop disease spread. John Snow, 1854. Identify cholera source in polluted London water supply. Advocated avoiding contaminated water to prevent disease. Joseph Lister, N67, developed antiseptic surgery to prevent microorganisms' entry into wounds. Explained milk souring as lactic acid fermentation, isolated Bacterium lactis. Introduced sterile surgery principles, known as the father of antiseptic surgery. Golden era. Louis Pasteur, 1857, early 20th century, till beginning of World War I. Discovered role of microorganisms in fermentation. Fermentation is the process that breaks down a substance into a simpler substance. Example: yeast. In 1857, Pasteur resolved the mystery of sour wine. He explained the role of yeasts in fermentation of fruits and grains, resulting in production of alcohol. Also observed that bacteria spoil wine. Louis Pasteur proposed germ theory of disease, and it also discovered anaerobic bacteria. He invented pasteurization to kill microorganisms in liquid and also involved mild heating to kill. Developed anti-rabies and anthrax vaccines. Identified cholera and rabies causative agents. Louis Pasteur's contributions. Discovered bacteria sensitivity temperature. Developed method to weaken bacteria, leading to vaccine development. Developed principle of immunization, weakened cultures. 1881, created anthrax vaccine and protected sheep against disease. Robert Koch. Proved studied germ theory of disease. Studied anthrax. Fatal disease of blood of sheep and cattle. Injected mice with diseased animal blood, observed consistent symptoms. Isolated rod-shaped bacilli from the blood of mouse. Found similar symptoms in oxen, isolated bacilli from their blood. Autopsied animals, found blood swarming with bacilli. Koch's postulates, methods to prone germ theory. Suspected microorganism present in diseased sick organism, but not healthy organism. Isolate microorganism in pure culture, on nutrient medium. Inoculate isolated microorganism into healthy host, the same disease must result. Re-isolate same microorganism from the experimentally infected host. 1880, Koch observed small masses of bacteria slice of potato which named colonies. Big group of millions of bacteria same type. Bois neck flask. Experiment and strictly disproved the theory of spontaneous generation. 1885, successfully immunized young Joseph Meister against disease rabies. But he never saw agent of rabies. Koch's isolated causative agents of TB, tuberculosis, typhoid, and diphtheria. Emil von Behring, First Nobel Prize in Physiology and Medicine for successful use of antitoxin in treatment of diphtheria. Other pioneers of microbiology, Kitasato and Koch, cultivated tetanus bacillus. Elie Metchnikoff gave term phagocytosis in 1884, eating of cells. Described body's defense process where WBC engulf and destroy microorganisms, won Nobel Prize in 1908. Notable contribution, Bacillus bulgaricus therapy and concept of health. Charles Nicolle proved typhus fever transmitted by lice. Albert Calmette developed harmless tubercle bacillus strain for immunization. Jules Bordet isolated pertussis bacillus. Developed complement fixation test for disease diagnosis. Ronald Ross awarded mosquitoes transmit malaria. Awarded Nobel Prize in 1982. David Bruce identified undulant fever cause. Associated tsetse flies with sleeping sickness. Masaki Ogata and Kiyoshi Shiga. Ogata reported that rat fleas transmit bubonic plague. Shiga isolated bacterium causing bacterial dysentery, intestinal disease. Howard Taylor Ricketts discovered agent of Rocky Mountain spotted fever. Daniel E. Salmon and Theobald Swith, American microbiologists, first who used heat-kill bacteria for immunizations. Salmon studied swine plaque, name linked to salmonella. Smith discovered Texas fever, cattle disease transmitted by ticks. Era of chemotherapy and microbial genetics. Paul Ehrlich and Sakahiro Hatu, 1910, discovered salvarsan for treating syphilis. Started the era chemotherapy targeting pathogens without harming the host. Gerhard Domak, 1935, found Prontosil against streptococci and staphylococci, and got Nobel Prize. Alexander Fleming, 1927, discovered penicillin from Penicillium mold. Penicillin became first antibiotic and is also known as wonder drug. Modern era. The modern era, significant roughly in 1970s, saw significant advancements in understanding manipulating microbial genetics. They cracked the genetic code, discovered cause of stomach ulcers, and faced new disease like AIDS and SARS, birds flu. Understanding microbes' roles in nature expanded, and we learned they're not all bad. They often help us out. In 2005, Barry J. Marshall and Robin Warren won Nobel Prize for showing that painful ulcers in stomach and intestine